Welcome to Advanced Exercise Physiology. This is Chapter 19, looking at the factors affecting performance. The objectives of this chapter are 1. To identify factors affecting maximal performance, 2. To provide evidence for and against the central nervous system being a site of fatigue, 3. To identify potential neural factors in the periphery that may be linked to fatigue, 4. To explain the role of cross-bridge cycling in fatigue, 5. To summarize the evidence on the order of recruitment of muscle fibers with increasing intensities of activity and the type of metabolism upon which each is dependent. 6. To describe the factors limiting performance in all-out activities lasting less than 10 seconds. To describe the factors limiting performance in all-out activities lasting 10 to 180 seconds. And to discuss the subtle changes in the factors affecting optimal performance as the duration of maximal performance is increased from 3 minutes to 4 hours. For those of you taking notes, here's an outline to follow the major concepts of this chapter. 1 would be sites of fatigue, 2 would be factors limiting all-out anaerobic performance, 3 would be factors limiting all-out aerobic performance, and 4 would be looking at the athlete as a machine. To start, here are the factors that affect performance. You can reference this in the book, but you can see that it includes diet, central nervous system function, strength and skill, environment, and energy production. Starting with the sites of fatigue, we see that fatigue is the inability to maintain power output or force during repeated muscle contractions. There is a central fatigue, which is the result of the central nervous system, and peripheral fatigue, which is a result of neural factors, mechanical factors, and energetics of contraction. Here is a diagram outlining all the possible sites of fatigue that can be referenced in your book. Central fatigue is the reduction in motor unit activation. It is also due to a reduction in motor unit firing frequency. The central nervous system arousal can alter the state of fatigue by the facilitating motor unit recruitment, which would involve increasing motivation or physical and mental diversion. Excessive endurance training, also called overtraining, reduces performance and leads to prolonged fatigue, etc. This can also be related to a brain serotonin activity. Looking at the quote-unquote central governor model, this is the conscious and subconscious brain and not the spinal cord or the motor unit. Now looking more closely at peripheral fatigue, we'll start with the neural factors. These involve the neuromuscular junction, not a site of fatigue. Looking at the sclerolemma and the transverse tubules, however, the abilities of muscle membranes to conduct an action potential, we see the inability of the sodium-potassium pump to maintain action potential amplitude and frequency. This can, however, be improved by training. In addition, an action potential block is seen in the T tubules. This is due to a reduction of the calcium release from the sarcoplasmic reticulum. In summary, increases in the CNS arousal facilitate motor unit recruitment to increase strength and alter the state of fatigue. Repeated stimulation of the sclerolemma can result in a reduction in the size and frequency of action potentials. However, shifts in the optimal frequency needed for muscle activation preserve force and output. Under certain conditions, an action potential block can occur in the T tubules to result in a reduction in the calcium release from the sarcoplasmic reticulum. Looking at other factors of peripheral fatigue, we will look at mechanical factors. One would be the cross-bridge cycling and tension development that depends on the arrangement of actin and myosin, the calcium binding to troponin, and the ATP availability. We also see that high hydrogen ion concentrations may contribute to fatigue. This would be due to the reduce of the force per cross bridge, the reduce of the force generated at a given calcium concentration, and the inhibiting of calcium release from the sarcoplasmic reticulum. We also see that a longer relaxation time is a sign of fatigue. This is due to a slower cross-bridge cycling. In summary, the cross-bridge's ability to quote-unquote cycle is important in continued tension development. Fatigue may be related in part to the effect of high hydrogen ion concentration and the ability of the sarcoplasmic reticulum to rapidly take up calcium. The end result may be a longer relaxation time, which affects the rate of muscle contraction. Finally, looking at peripheral fatigue and the energetics of contraction, we see the imbalance of the ATP requirements and the ATP generating capacity can lead to an accumulation of phosphate, which inhibits maximal force, reduces cross-bridge binding to actin, and inhibits calcium release from the sarcoplasmic reticulum. The rate of ATP utilization is slowed faster than the rate of ATP generation as well, and this maintains the ATP concentration. In summary, fatigue is directly associated with a mismatch between the rate at which the muscle uses ATP and the rate at which the muscle can supply the ATP. Cellular fatigue mechanisms slow down the rate of ATP utilization faster than the rate of the ATP generation to preserve the ATP concentration and cellular homeostasis. Now, looking at radical production during exercise and how it contributes to muscle fatigue, we see that exercise promotes free radical formation. These are molecules that contain impaired electron in the outer orbital. These are capable of damaging proteins, lipids, and DNA. What we see is that this can contribute to fatigue. What happens is there's damage to contractile proteins, both myosin and troponin, and this limits the number of cross bridges in a strong binding state. In addition, it depresses the sodium-potassium pump activity, which leads to disruption in the potassium homeostasis. In addition, optimal level of antioxidants can postpone this fatigue, but high doses can impair muscle function. Continuing with the energetics of contraction, we see muscle fiber recruitment and increasing intensities of exercise, where initially we have type 1 recruitment followed by type 2A and finally type 2X. Up to 40% of the VO2 max would contribute to type 1 fibers being recruited. Type 2A fibers are recruited at 40 to 75% of the VO2 max. An exercise greater than 75% of the VO2 max requires type 2X fibers. This results in an increase in lactate and hydrogen ion production, however. The following diagram shows the order of muscle fiber type recruitment that we just discussed as the exercise intensity increases. In summary, muscle fibers are recruited in the following order with increasing intensities of exercise: type 1, type 2A, and type 2X. The progression moves from the most of the least oxidative muscle fiber type. The intense exercise demands the type 2X fibers to be recruited, resulting in an increase in hydrogen ion production. Now, if we're looking at ultra short term performances, we are looking at events lasting less than 10 seconds. These are dependent upon the recruitment of the type 2 muscle fibers we just discussed that generate the greatest forces that are needed. This would involve motivation, skill, and arousal, which are all extremely important. The primary energy source during this is anaerobic, and we have the ATP creatine phosphate system as well as glycolysis involved. Creatine supplementation may improve performance in these short-term events. The diagram below will show factors affecting fatigue in these ultra short-term events. Please reference your book to look at these in more detail. In summary, in events lasting less than 10 seconds, optimal performance is dependent on the recruitment of appropriate type 2 fibers to generate the greater forces needed. Motivation or arousal is required, as well as the skill needed to direct the force. The primary energy sources are anaerobic, with the focus on phosphocreatine. Continuing with the short-term performance, again, these are events lasting 10 to 180 seconds as opposed to the ultra short-term performances. The shift is from anaerobic to aerobic metabolism, where 70% of the energy is supplied anaerobically at 10 seconds, where 60% is supplied aerobically at 180 seconds. Anaerobic glycolysis is the primary energy source during short-term performances. This results in an elevated lactate and hydrogen ion level, which interferes with calcium binding in troponin. What we see is the ingestion of buffers may improve performance in the short-term events. Here's another diagram looking at factors that might affect fatigue in short-term events. In summary, in short-term performances lasting 10 to 180 seconds, there is a shift from 70% of the energy supplied anaerobically at 10 seconds to 60% being supplied aerobically at 180 seconds. Anaerobic glycolysis provides a substantial portion of the energy, resulting in elevated lactate levels. Moving on to moderate length performances lasting from approximately 3 to 20 minutes, we see that 60% of the ATP generated aerobically is at 3 minutes. While moving up to 20 minutes, we see that 90% of the ATP is supplied aerobically. We also see that a high VO2 max is important. There's a high maximal stroke volume involved, as well as high arterial oxygen content that is involved with hemoglobin content and inspired oxygen. This requires energy expenditure near VO2 max, where type 2X fibers are recruited and a high level of lactate and hydrogen ion is accumulated. Now, looking at factors affecting fatigue and aerobic performance lasting 3 to 20 minutes, you will see below. Now, asking the question, is maximal oxygen uptake important for distance running performance? We see that VO2 max sets the upper limit for ATP production in endurance events, even though the race is not run at 100% of the VO2 max. Performance is also determined by the percentage of VO2 max at which the runner can perform, estimated by the lactate threshold, and running economy. In summary, in moderate length performances lasting 3 to 20 minutes, aerobic metabolism provides 60 to 90% of the ATP respectively. These activities require an energy expenditure near VO2 max, with type 2 fibers being recruited in addition to the type 1 fibers. Any factors interfering with oxygen delivery, for example altitude or anemia, would decrease performance since it is so dependent on aerobic energy production. High levels of hydrogen ion accompany these types of activities. Now moving to intermediate length performances, we see that these are events that last from 21 to 60 minutes. They are predominantly aerobic and usually conducted at approximately less than 90% of the VO2 max. A high VO2 max, however, is important. Other important factors are running economy, with a high percentage of type 1 muscle fibers, and environmental factors would include heat and humidity, as well as the state of hydration. Here is another diagram displaying the factors affecting fatigue and aerobic performances lasting 21 to 60 minutes. In summary, the intermediate length activities lasting 20 to 60 minutes are usually conducted at less than 90% of the VO2 max and are predominantly aerobic. Given the length of the activity, environmental factors such as heat, humidity, and the state of hydration of the subject play an important role in the outcome and performance. Finally, moving on to long-term performances lasting 1 to 4 hours, we see that they're clearly aerobic. Environmental factors become more important, as well as maintaining the rate of carbohydrate utilization, where we see that the muscle and liver glycogen stores decline, and the ingestion of carbohydrates is needed to maintain carbohydrate oxidation by the muscle. In addition, the subject would need to consume fluids as well as electrolytes, and diet also influences the performance. Here is another diagram displaying the factors affecting fatigue in aerobic performances lasting 1 to 4 hours. In summary, in long-term performances of 1 to 4 hours in duration, environmental factors play an important role as the muscle and liver glycogen stores try to keep up with the rate at which carbohydrates are used. Diet, fuel, fluid ingestion, and the ability of the athlete to deal with the heat and humidity all influence the final outcome and performance. Now, looking at factors affecting performance in ultra-endurance events, we see that examples would include a 166-kilometer mountain run, a triple Ironman triathlon, or a 24-hour run. The most important variables are the VO2 max and the percentage of the VO2 max that can be sustained. What we see are metabolic responses, in particular, fat oxidation is 3.5 times greater after the event. This is consistent with exercises less than 60% of the VO2 max. There's also a 60% reduction in muscle glycogen stores and the potential for hypernatremia. However, it only affects 4% of athletes. Finally, looking at the athlete as a machine, where we are continuing towards a goal to improve performance. The problem is the potential is to treat the elite athletes like machines, where they are a collection of parts evaluated by specialists, and there is a use of implementation of research to improve performance. However, this may be exposing the athletes to risk in research and or in implementation of techniques. It's important that an institutional review board is involved to minimize the risk to subjects being studied. That concludes the content for Chapter 19. Next are a series of study questions to help you test your knowledge. 1. List the factors influencing performance. 2. Is a limiting factor for strength development located in the CNS or out in the periphery? Support your position. 3. Tracing the path the action potential takes from the time it leaves the motor end plate, where might the weak link be in the mechanisms coupling excitation to contraction? 4. When fatigue occurs, there is still ATP present in the cell. What is the explanation for this? 5. Describe the pattern of recruitment of muscle fiber types during activities of progressively greater intensity and explain them. 6. As the duration of maximal effort increases from 10 seconds or less to between 10 to 180 seconds, what factor becomes limiting in terms of energy production? 7. Draw a diagram of the factors limiting maximal running performance of a 1,500 meter to a 5 kilometer run. 8. While at high VO2 max is essential to a world-class performance, what role does running economy play in a winning performance? 9. Give the lactate accumulation will adversely affect endurance. What test might be an indicator of maximal sustained running, swimming, or cycling speed? 10. What is the role of the environmental factors such as altitude and heat in very long distance performances of 1 to 4 hours in duration? This concludes Chapter 19 and Factors Affecting Performance. Please review the lecture and/or your text to further study or answer any questions. In addition, feel free to email me at any time. Thank you.